Hello, uh, good evening, good day, students. So today we will start with the blotting techniques, which I told you in the last week. I'll be dealing with. So blotting means it's a process of adsorption or absorption of the content by the paper, right? Generally, blotting is going to be that. So here, what is going to get absorbed or absorbed onto what? It is the absorption or adsorption of the nucleic acids, or it can be proteins, by the cellulose membrane um, or even the blotting membrane, and then using that particular membrane in order to identify the presence of particular type of either the nucleic acids, or even it can be a protein. So that is what blotting is. So blotting is going to be done with a it has some prerequisites, like we have to do an electrophoresis, so wherein we are going to be separating the nucleic acids, or we may able to in case if we are studying about the proteins, then we have to separate the proteins. So once they are going to get separated, then we are going to blot it, means we are going to transfer it onto the nitrocellulose membrane, and then we are going to use some labeled probes to detect those particular blotted components, and to facilitate the hybridization, uh, sorry and then we are going to study it, and either we can visualize. There are different types of blotting: southern blotting, northern blotting, western blotting, dot blot technique, and there is some people also call as southwestern blotting. Southern blotting, it's a method for identifying the DNA fragments uh, and containing a specific sequence, and that is done by using either DNA or RNA probes, which are labeled with radioactive labeling or fluorescent labeling. So the fragment containing the sequence can be visualized by autoradiography if it is radioactivity. Original developed by EM Southern, that's why people call it as Southern blotting. The procedure goes with, like, first the DNA sample should be means the one which we want. It should get electrophoresed after digesting it with the restriction enzymes. Then the bands on the gel are going to be denatured into single strand by using alkali, and subsequently we are going to lay it upon a particular nitrocell membrane by using buffers, and then we are going to blot it, means we will transfer that particular nucleic acids onto the um particular nitrocellulose membrane. That can be done by two ways. One is by through capillary action, so which will help in pulling it up, or we can use electroblotting, wherein the since nucleic acids are negatively charged, they are going to tend to move from means towards positive electrode. So what we can do is we can place the membrane at the positive side, and then we can allow, just like electrophoresis, we will allow the movement of this particular separated fragments of nucleotides within the gel onto the membrane. So once the transfer is done onto the membrane, then we are going to subject the membrane for fixing that particular nucleic acids to the membrane tightly by baking it at 80 degrees centigrade, and then after that the membrane is going to be taken and it is going to be dipped into the buffer containing the probes, which after incubation we can find that these particular probes, based on the sequence existence of their homology, they are going to undergo hybridization. And if there is any kind of hybridization, those particular DNA, those particular probes are going to be firmly interacted with the membrane. Those particular probes which are not involved in hybridization, they are going to be very loosely bound, which can be removed by simply by the process of washing by using the wash buffers. And later, after doing this particular washing 2 to 3 times, so that all the hybridized, means the one which is not hybridized, all those probes can be removed from that particular nitrocellulose membrane. So later it will be subjected for autoradiography, and then what people are going to do is they compare this particular position of the blotted position with the gel, so that the any DNA left can be means usually there will be two electrophotograms available, one which is used for blotting, the other will be kept as it is. So by and then what they do is they are going to measure the RF, and then they find the distance at which that particular blotted fragment is available on the nitrocellulose membrane, and similarly the position will be identified on the second electrophotogram, and then the DNA is going to be isolated and it can be used for further studies. The application of Southern blotting, it is extremely sensitive method for detection. People used in study for complex genome such as man, and when mini satellite probes are used, the technique can be used also for forensic purposes in order to detect the unknown DNA molecule. And the use of Southern blot technique is also done for analyzing the role of DNA methylation in gene expression. Blotting analysis is useful in the detection of mutated genes, identification of genetic disorders, and even for restriction analysis, RFLP, many more things. The next comes Southern blotting. Southern blotting, the difference between next is the northern blotting. The difference between the northern blotting and southern blotting is mainly the southern blotting mainly deals with the identification of the deoxyribonucleic acids, that is the DNA sequence, and all the work is done for the double stranded DNA molecule there, in identification of a double stranded DNA molecule. Whereas in case of northern blotting, it's all about identifying the RNA sequence. So here what we are going to be mainly doing is we will be separating the RNA molecule isolated from a particular host cell, and then these particular RNA molecules are subjected for size fractionation or separation by using electrophoresis. Then these electrophoresed RNAs are going to be transferred onto the nitrocellulose membrane. They'll be fixed with the nitrocellulose membrane, and then these particular fixed RNAs are going to be analyzed by using a specific probes, and these particular probes are going to be nucleic acid sequences, which can be of either single stranded DNA type, or it can be another RNA which is having complementary sequence. So this method was developed by Alvin Kemp and Stark, and they use a chemical reactive paper, that is diazotization of benzyl aminobenzoyl oxymethyl paper they actually originally used. As I told you, the procedure involves RNA of a particular species are going to be separated on the basis of electrophoresis using some denatured gel electrophoresis of either formaldehyde or glyazole, or even it can be with a dimethyl sulfoxide. Then separated RNA bands are going to be subjected for blotting, either through capillary blotting or through electroblotting. It will be transferred onto the nitrocellulose membrane. Then they are going to use a probes, that is DNA probes or complementary RNA probes, and then that hybridized position is going to be means, identified by autoradiography. So you can see how the whole procedure here. So we do electrophoresis, then the size fractionated components are going to be subjected for blotting, either through capillary blotting or through electroblotting, and then after blotting, that will be subjected, the membrane will be subjected for the hybridization in a hybridization chamber. Excess of means probes are going to be removed, all the unbounds are going to be removed, and the hybridized one are going to be subjected for. Then autoradiography will determine where exactly the blotting or sorry hybridization takes place. Application wise, it is used in the detection of and quantitative estimation of hybridized mRNAs, study of RNA degradation, RNA half-life, RNA splicing, and even useful in the study of gene expression. Next type of blotting is Western blotting. This particular technique is used to detect the proteins of a particular type. So till now what we have studied is about the blotting involved in the nucleic acids. One southern, which is DNA, then northern, which is RNA. Then now comes this is a protein identification methods, the Western blotting. So here what we do is, here the study is going to be done by in an indirect way about the capacity of producing the protein by a particular DNA fragment or a gene expression is going to be studied here. So people subject that particular cell for expressing the gene, and then the recombinant DNA which is expressed in the form of a protein is going to be studied by using Western blotting. So it's mainly used in the study of gene expression. Western blotting, it's a major application of the Western blotting. Even people also use it for the identification, qualitative, quantitative, all determinations people do by using this method. Procedure goes with, first we are going to take all the proteins from a particular cell or a particular protein. Then the extracted protein is subjected for separation by using polyacrylamide gel electrophoresis, and are then transferred into a nitrocellulose membrane by the process of blotting, which includes either capillary or electroblotting. Then these blotted membranes are going to be taken out from the blotting system, and these particular membranes will be subjected again for binding with a particular probe. So here is the key. Generally the probe which is used in case of Western blotting is going to be a protein again in nature, which is going to interact with the particular specific protein molecule. So here the interaction can be done, means it can be by the principle of immune reaction, where is an antibody and antigen interact and bind specifically with that particular antigen. So the antibody here will be generated against to a particular known protein or the protein of interest. Now in order to check whether the protein of interest is present and how much it is present, we can use this method where this particular antibody, it can be made before adding into the membrane. They're going to label this particular antibody by using the the enzymes, or that can be labeled with fluorescence, or even it can be labeled with a radioactive component. So if these particular antibodies are labeled with the enzymes, the enzymes are going to be, such kind of enzymes will be used which can convert a non-chromogenic substance into chromogen. Generally peroxidases, phosphatases, they are majorly employed here as an enzyme component. Or people use fluorescent dyes also, which is going to be used as a labeling component, or even radioactive components or radioactive elements. Now here this antibody which is labeled is added to that particular separated and blotted nitrocellulose membrane, and this, after addition of this particular component, so sometime people what they do is before adding the antibody, they can also add the blocking agents. The blocking agents which are going to specifically block all the areas which is not having the protein, means blotted onto it. Means nitrocellulose membrane, sometimes it can sterically, it can also interact with the antibody, it may bind with. Okay, so in order to avoid it, first all the surfaces where the protein is not there on the membrane will be first blocked by using some blocking agents. The most commonly used blocking agents are going to be the casein powders, so which is used as a blocking agents, which will go and bind with the nitrocellulose membrane except where exactly already blotted protein is present. Then after blocking, then they are going to add specific antibody which is against to the blotted protein molecule. And this particular antibodies, it depends. Some people you directly label this antibody, or some people are going to raise the anti-antibody against to the this particular antibody, which we can call it as primary antibody, the one which directly binds with the target protein. So now either primary antibody will be directly labeled, or it can be interacted, means made to interact with the secondary antibody which is going to be labeled. Either of the way, once the probe is added, so then if necessary second labeled secondary antibody will be added, or if not, they'll just use the labeled method in order to identify the presence of the desired protein. So if it is a conversion of a non-chromogenic into a chromogen, then they are going to use non-chromogens as a substrate, the enzyme which is attached to the antibody, which converts wherever the particular enzyme is present, there the product which is colored will get produced. For example, here there we can say uh, mm, there will be formation of blue color by the radius peroxidase when it's going to act upon a bromoethanol blue, so that is TMB. Then or else if it is going to be a radioactive element which is used, then autoradiography they'll go for. If it is fluorescent, they will watch for the fluorescence. So thereby they identify the presence of a particular desired protein molecule which is separated. So in case if let's say we want to use the particular protein and we want to work it on, so then there should be another electrophotograph which is run simultaneously along with the this first one. So then the one of it is going to be used for extraction process. If you come to the applications, it is being used in as a confirmatory test in case of HIV, where people are using it as in order to identify the antibodies present in the patient's blood sample. Then the Western blot, Western blot is also being used as a definitive test for in case of BSE, bovine spongiform encephalopathy. People are also using it in the identification of hepatitis B infection, and in many more other kind of infections in the form of diagnostics, or in the form of for the study purpose in case of research, are all being used. uh, Means they're using the Western blotting. So the last type of blotting technique is dot blot assay or dot blot technique. The technique is used to detect the presence of a given sequence of DNA or RNA in a non-fractionate DNA sample, means which is not subjected for separation by size. So here DNA in many samples can be tested in a single test. So that's the beauty here. So it is not having means and restrictions like generally which is existed within the electrophoresis, or within the southern hybridization or northern hybridization, where particular type of DNA or RNA can only be identified. So in this method we can subject means we can analyze DNA or RNA isolated from many number of organisms or species. So the presence, this method is simply represents like it's a simplification of northern blotting or southern blotting, or even it can be a western blotting method. In the dot blot, the biomolecules to be detected are not first separated by electrophoresis. Instead, the mixture containing the molecule is going to be applied directly on the membrane as a dot. Then the spot through circular template is going to be directly made onto the membrane, followed by the detection by either nucleotide probes or the antibody probes as in western blotting. If you check into the procedure, sample DNA or RNA and or even uh, the proteins are separate are going to be taken, and it will blotted onto the nitrocellulose membrane as a dot. Then the if it is DNA, the denaturation is going to be done by baking the membrane to 80 degrees centigrade. Then the filter is treated. There is a membrane is going to be treated with appropriate radioactive signal standard protein probes under a particular condition of hybridization. Then they are going to wash off the filter paper, remove the unhybridized probes. The dots with hybridized probes are detected by the autoradiography. And what is the intensity of the dot can also be used in identifying the quantitative determination can be done. So you can see how the dots are going to appear in this method. This is dot blot assay, which is very widely used. In terms of usage, it is similar to the southern or northern blotting. Only thing is this method is used uh, with a very simplicity and rapidity. That's it. Thank you. So I'll continue later with the applications of recombinant DNA technology, wherein we'll discuss about how people are actually using this method in producing some recombinant proteins which are being used for therapy and other things.